And at the heart of what I've done over the last, well, certainly the last 25 years, is to use one of the great technologies of the 21st century, which is genetic engineering. This is the protein sequence of the protein that makes the light in the British glowworm that we cloned. We isolated the DNA just from half a dozen, so we didn't damage the ecology of the glowworm. And we got the sequence out from the DNA sequence. And when you teach about genetic engineering, I'm sure you talk about the genetic code and the letters, the one-letter code and the three-letter code. So, like all proteins, it starts with methionine and it stops here at 547 amino acids. I'm not here to promote planting of GM crops, but I am here to promote the importance of genetic engineering. It's got huge economic potential. We're not doing enough of it in Wales. Biotechnology, you could make very, very small amounts of material without having pulped all them. And it's worth billions, not just millions, it's worth billions. If you took the human insulin gene and you put it into a potato plant and you grew it in a 10-acre field at the bottom of my grounds in Pembrokeshire, the value of the potatoes to the farmer would be worth about £10,000. If it had human insulin in it, you could make enough insulin to supply the whole of the British diabetic population for 1 year. The current cost is £800 million. That is the value of biotechnology. So one of the things that I've done is to engineer these proteins, and you'll see in a minute why we've done that. Now, discovery. We talked about glowworms and fireflies. There are earthworms, there are the New Zealand glowworm, there are fungi that make light. But the real place you look for bioluminescence is in the sea, and particularly the deep sea. So as I said earlier, this is the ecology of the dark. It's the biggest ecosystem on this planet. Half of the Earth's surface is covered by sea 1,000 meters deep. There, virtually everything is luminous. So I was lucky enough to go out on research cruises to collect these animals. This is Madeira. This is the American research ship. This is the British ship here. No, not really. This is the ROS Discovery, which has been scrapped now. They had special nets to collect deep sea animals. And here are some of the luminous animals that I collected. Fish, squid, jellyfish, fire bodies, shrimp, euphorsids, the krill, the whale eat, and more invertebrates like copepods and ostracods. All of these are luminous. This fish is luminous. It doesn't have a common name, and it's probably the commonest vertebrate in the whole world. That's how important bioluminescence is in the sea. It's the communication system in the biggest ecosystem on our planet. And we can start to then see, why is this shrimp red? And I showed kids, they say, well, it's not red, it's orange. Well, why is it orange or red? And these are the choices. It's being cooked. It's being painted red. It isn't red. I don't know. How many people think it's being cooked? Well, obviously, and anybody know what the answer is? We've got the sort of reason, but the real reason is it isn't red. I've held it in my hand, it squirted out blue luminescence. In the deep sea, all the light is blue light. And although there's rather too much light in here, but when I've got a dark room, I'm very keen on demonstrations. I'm hoping to have a bit more time and develop booklets for teachers to do live demonstrations. So if you make a model, this is obviously it's too bright in here, but if you shine blue light onto a red object, what colour is it? You ask your pupils that. If you shine blue light on what we see as a red object, what colour is it? Thank you. It's black. This is nature's deep sea black pigment, because there's no red light there. So colour is based on reflecting the light which is not absorbed. But if you shine blue light, there is no red component, so it just absorbs all the blue light. But if I shine red light onto it, you can see that it is red. Of course, it doesn't show up too well in this room. Unfortunately, for this shrimp, there's a group of fish there which are called dragonfish, which do emit red light. This is its mouth, these are its teeth. It's about that long. Watch it, when you put that in the catch, you get a bit long. This is his eye. There's a blue light organ here and a red emitting light organ here. The red shrimp, or the black shrimp in blue light, can't see red light. But this fish does have pigments which see red light, so it has an invisible torch which lights up the red shrimp, so we can eat it without the shrimp knowing what's happening. We now start to see some of the important discoveries and have led into big markets. The lady with the lamp, Florence Nightingale, of the Crimean War. And she observed something very remarkable about bioluminescence. There are fish which contain luminous bacteria, which glow. They flash by having a flatter skin. But there's a species which lives in the soil. And there are many famous descriptions of it infecting bodies on battlefields in the American Civil War, in the Chinese literature. But what Florence Nightingale saw was that when she walked down the wards at night, some of the wounds were glowing, amazing, blue. And so she asked the orderly, "What is that?" And they said, "Well, yes, ma'am, we've noticed something very interesting, because the soldiers that have glowing wounds don't get gangrene. So we've been transferring the light to other wounds, and they don't get gangrene either." And there's a slightly apocryphal story, because I've been through hundreds of Florence Nightingale's letters, and I can't find the exact reference, but the story is true, because quite recently the genome of the bacterium was sequenced, and it turns out that this bacterium produces a very potent antibiotic. So this was probably the first use of antibiotics, well, 60, 70, nearly 100 years before Fleming and Laurie and Jane developed penicillin. So these are light-emitting bacteria. They were also used at the turn of the 19th, 20th century to show that plants make oxygen, but they also led to another very, very important discovery in microbiology. Because if you take one bacterial cell, it doesn't make light. Why should it? You can't see it. But if you grow it up into a colony, it does make light. And this is because each individual cell produces a very small amount of substance called an autoinducer. So each cell secretes a little bit, it adds up. When you have a quorum, all the genes switch on in all the cells and you can see the light. Now, a very good friend of mine in Nottingham University, who very sadly died about 10 years ago of brain tumour, he thought, well, this can't be unique to luminous bacteria. If he took the substance which makes the gene switch on in the luminous bacteria in the fish, or the fish, and he added it to a human pathogen, and it switched them on. So it turns out that a very, very important part of pathogens is they have to be a quorum. They then switch on all their genes, for example, to produce toxins or to grow faster. It's a very, very important principle in microbiology.